So in this lecture, we are going to focus on chromosomal and gene mutation detection. Now, we've already talked about chromosomal mutations and gene mutations in general. So we're really going to focus on how to detect these types of mutations that may occur. So we're really focusing on the technique here. At the end of this lecture, you should be able to state the karyotype of a normal male and female, identify the chromosomal abnormality in a given karyotype, compare and contrast both interphase and metaphase FISH analyses, discuss the various methods used to detect both chromosomal and gene mutations, and describe mutation nomenclature for chromosomal and nucleic acid alterations. So we already went over the different types of mutations. You have genomic mutations where you have abnormal chromosome numbers, and this results in monosomy, polysomy, and aneuploidy. We also have chromosomal mutations where you have an abnormal chromosome structure, or there are gene mutations where you have changes in the DNA sequences within specific genes. And remember your chromosome morphology, where you have your telomeres, which are your chromosomal ends. You have your centromere, which is in the middle of the chromosome. It's the site of spindle attachment. You get constriction of the metaphase chromosome at that center portion, and that's where you define your two chromosomal arms, the P and the Q. You also have your nucleosome, where your DNA double helix is wrapped tightly around your histone proteins. So defining your chromosomal location, you have your P arm of your chromosome, your Q arm, your centromere, which is directly in the middle, the pinched in area. You have your various regions of the chromosome. You also have your banding patterns and then your sub-bands. So the arrow on this slide is pointing to chromosome 17, the Q arm at the region 1, band 1, and sub-band 0.2. So sub-band 2 there. So if you're reading a chromosomal location, that's how you need to read it. The first number is the chromosome, then the arm, then the region, the band, and then the subband. So you get chromosome morphology changes during cell division. So you have your DNA double helix, and changes may occur during interphase, the G1S and G2 phases. You get your chromatin or beads on a string that will develop. You get your chromatin in your nucleosomes, and changes are going to occur in metaphase during mitosis. You have your extended metaphase chromosomes and condensed metaphase chromosomes. So your chromosomes are constantly changing as they divide. And remember, you have your cell division cycle. We talked about mitosis and meiosis in one of the earlier lectures. So you have your interphase, you have your G1, your S, your G2, as well as your mitosis. And in mitosis, you have prophase, anaphase, metaphase, and telophase. And your morphology changes are usually going to occur in a chromosome during interphase or during the metaphase of mitosis. So in identifying chromosomal mutations, you have to visualize your chromosomes. So conventional cytogenetic stains are used to visualize chromosomes, and those include Feulgen's, Wright's, and hematoxylin stains. However, now fluorescent stains are more commonly used, as well as chemical dyes that react with specific regions in the chromosome, and that results in banding. So we talked about banding briefly in a previous lecture when we were talking about chromosomes, but we're going to talk about it in slightly more detail. Q banding is when you have chromosomes that are stained with fluorescent dyes such as quinacrine and quinacrine mustard. And Q banding is going to give you an intense staining of the human Y chromosome. Your G bands or G banding is when you stain with Giemsa stain, and these give you patterns similar to what you would get in Q banding. So the appearance of your banding patterns depends on the treatment of the chromosome before you stain with your Giemsa stain. You can also do high resolution banding, where you can increase the number of bands that you could see by staining the chromosomes before they reach maximal metaphase condensation. There are also R bands and C bands. So R banding, you would do a very harsh treatment of your chromosome before you do your Giemsa stain, which produces a pattern opposite to the G banding pattern. R bands can be visualized after staining with acridine orange. C banding, you would do an alkali treatment of your chromosome, and this is going to result in staining of the centromere. So here's a schematic of your banding patterns of your chromosomes. And you have your Giemsa banding or your G bands. You have your C bands, your centromere banding, or your reverse bands, which is your R bands, which are the reverse of the G banding pattern. And these are stained metaphase chromosomes. You can also use other stains to visualize chromosomes, such as nuclear organizing region staining or NOR staining, where you treat chromosomes with silver nitrate, and this is going to stain the constricted regions on acrocentric chromosomes. You can also do DAPI staining, and this is going to bind to surface grooves of double-stranded DNA, and it fluoresces blue when you expose to ultraviolet light. So this is going to be used to visualize chromosomes as well as to visualize entire nuclei. There are other ways to detect genomic and chromosomal mutations, and these include karyotyping as well as fluorescence in situ hybridization or FISH. And there are two different types of FISH, interphase and metaphase. So karyotyping. A karyotype is the complete set of chromosomes in a cell. Karyotyping is when you directly observe the metaphase chromosomal structure by arranging the metaphase chromosomes according to their size. So it requires collection of living cells, growing these cells in tissue culture for 48 to 72 hours. Then cell division is stimulated by adding a mitogen, such as phytohemagglutinin. Cells are then arrested in metaphase by adding coulsamide, and this is an inhibitor of microtubule formation. So the chromosomes yield what's called a chromosome spread, where the cell nuclei are going to be disrupted with a hypotonic saline, so they're going to be broken open, and then the 23 pairs of chromosomes are then assembled into the karyotype according to their size and centromere placement. So there's an international system for human cytogenetic nomenclature, or ISCN. So this nomenclature is very important. So for example, 46, XX indicates a normal female. 46, XY indicates a normal male. And you can identify your G-banded chromosomes and G-banding patterns. So here, this is an example of a normal female karyotype, and we did see this previously. So 46XX. So you see your 23 pairs of chromosomes. You see that there are 2 X chromosomes and no Y chromosomes there. This is your female karyotype using the high resolution G banding, so you could really see those bands more distinctly with the high resolution staining. This is a karyotype indicating aneuploidy. So you have 48, XXXX. So instead of only having two X chromosomes, you have four there. This is a karyotype indicating trisomy 21, where you have 47XX, so this is a female, plus an extra 21, chromosome 21. So you could see at the 21 location, you have 3 instead of 2. You can use karyotyping to look for translocations. So remember, there are reciprocal translocations and balanced translocations. So in reciprocal translocations, that's when parts of two chromosomes exchange material. So each chromosome is going to break, and then those broken chromosomes are going to recombine or reassociate with one another. So it doesn't necessarily result in a gain or a loss of chromosomal material. Your balanced translocation, these can occur without any phenotypic effects. So balanced translocation that occur in germ cells can eventually become unbalanced if they don't assort properly during the mitosis, during cell division and mitosis. Okay, this is a karyotype looking at a balanced translocation. So you have 45, XY, 1X, 1Y, this is a male, the little t, which indicates a translocation. And if you look at chromosome 14 in the arm Q, you can see that that chromosome is abnormal. So there's been a translocation in that area. There's an arrow pointing to 21, band Q. So there's been a translocation between chromosomes 14 and 21 in the Q bands. You can also look at other types of chromosomal mutations using karyotyping. So you could see deletions, especially larger deletions that may cover millions of base pairs. You could see insertions, inversions. You could see derivative chromosomes where you would have an abnormal chromosome consisting of a translocated or rearranged part from two or more chromosomes that are joined up with a normal chromosome. So after you do your karyotyping, you want to write up the results. So karyotyping is expressed as the number of chromosomes per nucleus. So normal is going to be 46, followed by your sex chromosomes, and it's going to be XX if it's a female or XY if it's a male. And then you're going to follow that with your genetic abnormality. So if you had, for example, a deletion in the long arm, which is Q, of chromosome 7 at region 13, it would be 46, XX because you have a female. DEL meaning deletion, your parentheses 7, which is the chromosome 7, and Q13 meaning the long arm of region 13. If you have a translocation between the short arm of chromosome 5 and 17 and region 13, band 3 and region 13, that would be 46XY, T for translocation, 5 semicolon 17, and then your P13.3 semicolon P13. So now we're going to discuss fluorescence in situ hybridization, or FISH. So why would you use FISH? And the FISH is used to identify and characterize numerical and structural chromosome abnormalities. You can detect microscopically invisible deletions. You can also detect sub-telomeric aberrations, and it's also used for prenatal diagnosis of common aneuploidies. So FISH is going to detect protein, RNA, and DNA structures that are in place within the cell, which is why it's called in situ. It's a common method used in cytogenetic studies. So fixed cells are exposed to a probe, and usually the probe is going to be a 60 to 200 kilobase fragment of DNA that's covalently attached to a fluorescent molecule. The probe is going to hybridize to its complementary sequence within the cellular nucleic acid material. So some common probes used in fish. You can use chromosome-specific centromere probes, which are going to hybridize to the centromere region and detect aneuploidy in both interphase and metaphase. There's chromosome painting probes, which these probes will hybridize to whole chromosomes or regions and will characterize chromosomal structural changes during metaphase cells, or unique DNA sequence probes, which are going to hybridize to unique DNA sequences and detect genetic rearrangements, deletions, and amplifications. You can also use telomere-specific probes, which are going to hybridize to the sub-telomeric regions and detect sub-telomeric deletions and rearrangements. So depending on what regions the probes bind to, you're going to be able to detect any alterations in that region of the chromosome. In interphase fish, the bound probe can be visualized under a fluorescent microscope in the nucleus of the cell. So you're actually looking at the nucleus. The probes are designed to be specific to a particular chromosome or chromosome region so that the microscopic image will correlate with the state of that chromosome or that region. So for example, if you were to probe any unique region on chromosome 22, you may yield an image of 2 signals per nucleus, and that's going to reflect the 2 copies of chromosome 22 in a normal somatic cell nucleus. If you had a deletion or a duplication of the region, your probe then is going to give you only 1 signal if there's a deletion, or more than 2 signals if you have a duplication. So you have your hybridization of your complementary gene or region specific probe to your chromosome, and this can occur during either interphase or metaphase cells on your slide. So if you have that complementary region to whatever your probe is then going to bind, and you're going to be able to see that signal within the nucleus under a fluorescent microscope. So you can use interphase fish in these different probes to look for genetic abnormalities. So if you have greater or less than 2 signals per nucleus, that would be considered abnormal. So a normal signal is going to give you 2. So in your cell nucleus, you're going to see 2 signals. Abnormal, you would either see 1, which would either indicate either monosomy or deletion, or more than 2, which is going to indicate trisomy or an insertion. So once again, you're looking at the nucleus, you're looking at your probes that are going to hybridize, and if a normal diploid cell is going to give you 2 signals, triploidy would give you 3, a deletion would give you 1. You can also look for structural abnormalities using fusion probes. So your probes are going to fuse to or hybridize to specific regions in your chromosome, and you might see a translocation where you would see 2 signals on a chromosome where you would normally only see 1, or maybe you wouldn't see a signal on a chromosome if it was swapped out with a different region so your probe doesn't bind to it. So you can see many different types of abnormalities using these probes. You can also use break apart probes, and these probes contain a break point in them. So normally these probes would bind together in a specific chromosome. However, if there were a translocation, those probes are not going to bind together because you have a different region in that chromosome due to the translocation. In metaphase fish, you would prepare your chromosome by culturing them for 72 hours. 45 minutes after you harvest, you would add your coulsamide to arrest your cells in metaphase. You then suspend your cells in a hypotonic medium and you fix your cells with a methanol acetic acid reagent. The fixed cell suspension is then applied to an inclined slide and allowed to dry. Then you do a second treatment with 70% acetic acid, which sometimes improves the chromosome spread. In metaphase fish, using a phase contrast microscope, your chromosomes should appear well separated and they should have sharp borders, and also the cytoplasm should not be visible. This is a schematic of what a translocation using metaphase fish and whole chromosome painting would look like, and you can clearly see that a translocation has occurred using this method. You can use these methods to do comparative genome hybridization, and this is going to allow you to detect intrachromosomal amplifications or deletions. So what you do is you take DNA from both test and reference samples, and you're going to label them and use them as a probe on your normal metaphase chromosome spread. So the dyes that you're going to use are cyanine dyes such as Cy3 and Cy5, and these are going to be your fluorescent labels for your test and your reference DNA. So Cy3 fluorescence at a wavelength of 550 and is usually green. Cy5 fluoresces in the far red and usually is red. So your DNA to be tested is going to be partially digested with DNase, and that's going to produce fragments. So you're not going to do full digestion but partial digestion. So that's going to bind efficiently to your denatured DNA in your metaphase chromosome spread. So in comparative genome hybridization, you would have a test sample compared to a normal reference sample on a metaphase spread. So normally your test and your reference signals are going to be equal. However, a higher test signal would mean that there's been an amplification, and a higher reference signal would mean that there's a deletion, and that's what this slide is showing. So how do we detect gene mutations? So we talked about detecting chromosomal mutations. So now we're going to move into gene mutations. And we're going to talk about mutation nomenclature. So when we're talking about nucleotides, we start out with the base position and then whatever the mutation is. So in the first example, base position 5162 in that gene, the original base was a G and it was converted to an A. And you're going to do this, you always start out with the base position and then whatever the mutation is. If you're talking about the protein, you start out with the original amino acid, the position, and then the replacement. So within that protein, you should have one amino acid at that position, but it was replaced with a completely different amino acid. So that's how you're going to look for your mutation nomenclature in genes. And like I said, it could either be the nucleotide sequence or the amino acid sequence, depending on if you're looking at the DNA or at the protein. So we already talked about point mutations, where you have mutations that are involving one or very few base pairs. Usually these are not detectable by the cytogenetic method, so that's why you have to look at the gene methods. So these point mutations are only detectable at the DNA sequence level. So remember that point mutations don't always have an effect on the phenotype, and there's many different point mutations. We've already talked about all of these, where you have silent mutations, nonsense mutations, frame shift mutations, etc. So the types of mutation detection methods in genes, there are hybridization-based methods, sequencing-based methods, and cleavage-based methods, and we're going to talk about a few of these. The first one we're going to talk about is single-strand conformation polymorphism, or SSCP. And this is going to scan several hundred base pairs, and it's based on intrastrand folding. So single strands will fold according to their sequence. And even if there's just one nucleotide difference, that's going to affect how it folds. So your folded single strands are called your conformers, and these are resolved on a gel, and how they move through the gel is going to be based on their size and the shape. So you're going to use polyacrylamide gel under a controlled denaturing and temperature condition. And the speed of that migration is based on the size and the shape. So differences in shape are caused by sequence differences. And you could look at these bands using silver staining, radioactivity, or fluorescence. So this is a schematic. You amplify your region to be scanned using general PCR. You have your normal control and you have your sample, and maybe that sample has a mutation. You then are going to denature your products, and these are going to fold up according to their sequence. Then you're going to separate them using polyacrylamide gel or capillary gel electrophoresis. So if you were looking at polyacrylamide gels, you would see your normal control and you would see the size and location of that. And then if you had a mutation, there's going to be a difference in folding, so it's going to run through the gel differently. So you're going to see two different bands, or however many bands you happen to be looking at, but it's not going to run the same as your test control. If you had a mixture of your control and your mutation, you're going to see your normal bands, and you could see the altered band locations because of the mutation. The same thing, if you're looking at capillary gel electrophoresis, you're going to get a difference in the location of the peaks, whether you have normal or a mutation. Here's an actual polyacrylamide gel that's been stained with silver stain. You could see your T1 is your test sample, no mutations, two nice crisp bands. And your T2 would be your sample with your mutation, so you can see there's an extra band in there. And then your normal control. So the SSCP method has been reported to detect between 35 to 100% of possible mutations. It's actually sensitive enough to detect mutations in a sample that has as low as 5% of mutant cells. However, the more mutant cells you have in your specimen, you're going to get better results. So if you have at least 30% mutant cells in your sample, you're going to get pretty reliable results. So if you have an inherited mutation, 50% of your cells are going to have that mutation. So you're going to be more likely to see that mutation using the SSCP method. Somatic mutations, however, are mixed with normal cells. So it really depends on how many mutant cells and how many normal cells that are present. If there's more mutant cells, you're going to be more likely to see that mutation. So another method, we already talked about this method very briefly when we talked about gel electrophoresis. So this is denaturing gradient gel electrophoresis, or DGGE. And what this does is it detects differences in denaturation between a normal and a mutant DNA molecule. And this can actually detect as little as a 1 nucleotide sequence difference. So you have double-stranded DNA fragments that are between 200 and 700 bases and lengths, and they are prepared using PCR amplification or restriction digestion to break your DNA molecule up into pieces. These fragments are separated on polyacrylamide gel, and this polyacrylamide gel has a gradient of concentrations of urea and formamide. And urea and formamide are denaturation solutions, so they're going to denature double-stranded DNA. So as those double-stranded DNA fragments move through that polyacrylamide gel, the denaturing conditions increase as it moves through the gel. So the complementary strands, or strands, the double strands, are going to begin to denature and become single-stranded. So in your denaturing gradient gel electrophoresis, you're going to form single-stranded DNA as the DNA moves through the denaturing gel. And as you get your single-stranded DNA, the migration is going to slow down. So a one nucleotide difference between two DNA molecules is going to cause both of those molecules to denature at different positions in the gel. Now we're going to go through allele-specific oligomer hybridization, or ASO. And this is actually a dot blot method. And we talked briefly about dot blots when we were talking about blotting technology. So this is going to utilize the differences in melting temperatures of short sequences of approximately 20 bases with one or two mismatches, and those that have no mismatches. So it really relies on the binding effects of nucleotide mismatches. So you have your specimen in your solution and it's spotted on your nitrocellulose membrane, or making your dots. Then you have your labeled oligonucleotide probe that is going to hybridize to your dotted specimen on your nitrocellulose membrane. So this slide shows an example where you have 3 specimens that you have spotted or dotted on 2 nitrocellulose membranes. One of the membranes is exposed to a probe that you've generated that's complementary to the normal sequence. The other membrane is exposed to a probe that's complementary to the mutant sequence. So wherever you have normal sequence, that's going to light up. Your dot is going to light up when you use your positive probe. Wherever you have mutant sequence, that's going to light up where you have your mutant probe. And so you could use this dot blot method and these various probes to know if you have a mutation or not. So this slide shows you an actual dot blot using a chromogenic probe. In your first lane, you have a normal sample, so you're only going to see your dot spot light up when you have your normal probe and not your mutant probe. Number 2 is a heterozygous, so you're going to expect to see the sample light up in both the normal probe and the mutant probe. M is heterozygous mutant control. Again, you're going to expect to see it light up in both the normal probe and the mutant probe. The positive lane is a normal control, and you're only going to expect that to light up when you use the normal probe and not the mutant probe. And N is the negative control, and neither one should show anything. Another method to detect gene mutations is using Melt curve analysis. So remember, the nucleotide sequence has an effect on the melting temperature, and you can determine the melting temperatures based on the sequence. So the Melt curve analysis can be performed with or without the use of probes. So what you do is you take your double-stranded DNA-specific dye, such as ethidium bromide or cyber green. You can also use FRET probes. So your double-stranded DNA-specific dyes, such as your cyber green, are only going to fluoresce when they're bound to double-stranded DNA. So if you denature your DNA to single strands, you're not going to get fluorescence because the cybergreen will only bind to double-stranded DNA. So every sequence has a specific melting temperature based on those nucleotides. So you can do what's called a melt curve, and that's going to indicate what sequence is present. So your melting curve, as you increase your temperature, depending on what sequence is there, whether it's homozygous, mutant, or normal, or heterozygous, the nucleotides are going to differ between those situations, and your curve is going to differ as you increase the temperature. So those strands are going to melt at different temperatures because their nucleotide sequences are different. So you can actually use a computer software, to convert the melt curve to a derivative of fluorescence. And then you're looking at these little peaks, which are going to tell you if you have a heterozygous, a normal, a homozygous, etc. So we have array technology, and we're going to first talk about reverse dot blot methods. So your regular dot blot methods are like we just talked about with the ASO, where you're going to immobilize your specimen to a nitrocellulose membrane and then hybridize your probe to it. Well, reverse dot blots are the opposite. You immobilize your probe, either mutant or normal probe, to your membrane, and then you're going to amplify a sequence and expose that to the immobilized probes, and only the exact complementary sequence is going to hybridize to your immobilized probes. So if you get color, that's going to indicate the binding to the test DNA to normal or mutant probes. So this is going to be used to investigate multiple genomic sites simultaneously. So instead of investigating one gene and another gene and another gene, you can look at many genes at the same time. So you have your unlabeled probes bound to your substrate, you label your DNA, and then you're going to hybridize your labeled DNA to your immobilized probes. There are many different types of array technologies. There's microarrays, there's high-density oligonucleotides tide arrays, there's microelectronic arrays, and the detection systems are going to vary depending on the array that you're using. Also, the substrate is going to vary, whether it's going to be glass or an electrode grid or nitrocellulose membrane. You can use microarray technology to do your comparative genomic hybridization, where you're going to detect genomic amplifications and deletions. You can use it for expression analyses, and this you can detect changes in gene expression, looking at variations in messenger RNA. You can do microarrays to detect single nucleotide polymorphisms, and this would be a high-density oligonucleotide array and looking at single base differences in your DNA. So your high-density oligonucleotide arrays. So you can look at thousands of genes all at the same time. So what you would do is you would have an array for each sample that you wanted to analyze. Your unlabeled probes are then bound onto your substrate. And usually these probes are going to be between 10 and 25 nucleotides in length. So these are your oligomers, so they're 10 to 25 mers sitting on your substrate. So then your test DNA is going to be fragmented before you hybridize. So you're going to expose your DNA to an enzyme to cut it into pieces. So these short pieces are going to bind only to the complementary sequences that are immobilized on your array. So you can do tiling or overlapping probe sequence to do a blanket detection of nucleotide changes in the sample. So you can use various probes on the same array, and so that you can look at many different types of nucleotide variations all at the same time. So what you're going to get is a fluorescent signal when you have your hybridized DNA to the probe. And when you see your fluorescence, that fluorescence has to be normalized or averaged using computer software. So it's only going to indicate fluorescence if over a threshold level has been generated. So here's an example of your high-density oligonucleotide array, and usually this is going to be in a graph. So on the top you have your normal sequence, on the bottom you have a heterozygous sequence, and this is representative of 5 probes, and each probe has the indicated base or the deletion. So you could see in the bottom you have a different peak in the middle than you do on the top, and that's going to indicate your heterozygosity. Another method is sequence-specific primer PCR, or SSP-PCR. So PCR primer extension requires that the 3' base of the primer is complementary to your template, so that you have your normal template, you have your primer, and that last base, a 3' base, you have a G, which is complementary to a C that's in the sequence. However, if you have your mutant template, there's a T instead of a C, so you're not going to get any amplification because your primer is not going to be able to hybridize and amplify because the last base is different. So you have to design your primers for this method very specifically. So you're designing your primer to detect specific mutations in your DNA. So generation of a PCR product indicates the presence of the mutation or polymorphism in the template. So if you design your primer so that you're going to see a specific mutation, you're only going to get amplification when you have that exact mutation. So here's an example of your SSP PCR. And in this example, you have a C to T mutation. You have 2 different primers. You have a longer primer that you designed with the mutated complementary nucleotide. So the mutation is a T, so you design it with your A at the 3' end. So since this primer is a longer sequence, it's going to give you a larger amplicon. You have your other primer, which is specific for your normal sequence. So you can look at the products and you can distinguish them from their size. So if you run your gel with your PCR products, you have your molecular weight marker. Then in the second lane, you have your normal sample, which only is going to amplify with the smaller primer. So you get a small amplicon. Your third lane is a heterozygous, so you're going to get the larger amplicon and the smaller amplicon. In the fourth lane, you just have your homozygous mutant, and since the mutant was a longer primer, you're only going to get your larger amplicon. Another method is allelic discrimination, which uses fluorescently labeled probes. This is similar to the TACMAN technology for real-time PCR. So you generate a color signal for your mutant or your normal sequence, so a different color. So this is performed on your real-time PCR instruments. You have your probe that's complementary to your normal sequence that's going to be labeled with one color of dye. You have your probe that is the complementary sequence to the normal sequence that's labeled with a different colored dye. And depending on what color fluorescence you get, that's going to indicate if you have your normal sequence or your mutant sequence. So So your signals are detected and analyzed through the computer software, and you can look at multiple samples at the same time. So you can look at 10 different samples with the 2 different probes that are different colors, and you're going to see what fluorescence that you get and know if you have a mutation or not. Another method is restriction fragment length polymorphism, and this method is a southern blot. So RFLPs use the southern blot methodology. So it's based on restriction enzyme site recognition to detect sequence changes. So if you have an alteration in your sequence, you are going to either delete or generate possibly an restriction endonuclease sequence site. So for example, in a specific region of DNA, you have a G to EA mutation. And on the top, you can see your normal sequence, and on the bottom, you can see your mutated. In red, you could see the A. So you have a G to A mutation at that location. And it actually generates a BAMH1 site. GGA-TCC is the 6 nucleotide sequence that the restriction enzyme BAMH1 recognizes. So when you have a mutation in that exact location, you generate a BAMH1 site. So when you take your DNA and you cut it with BAMH1, you're going to get a cut there, so you're going to get a different banding pattern on your gel. Another method is heteroduplex analysis. So in order to form a heteroduplex, you have your double-stranded DNA duplexes that are complementary to one another. If you heat various double-stranded DNA duplexes, they're going to denature and become single-stranded. Well, if you slowly cool those single-stranded DNA fragments down, they are going to hybridize, and they won't necessarily hybridize to their exact complementary other half. So you're going to get double-stranded DNA that are not identical complementary sequences, and that's what a heteroduplex is. So your heteroduplexes are going to migrate differently than homoduplexes through an agarose or a polyacrylamide gel. So you can analyze your heteroduplexes using single-strand specific nucleases. So nucleases are enzymes that cut single-stranded bubbles that occur in a heteroduplex. So you can amplify your region of interest using polymerase chain reaction. Your amplicon is going to be denatured and then renatured either with or without adding your normal PCR product. So those renatured duplexes are then digested with your enzyme nuclease, and then you look at your products using gel electrophoresis. So here you have double-stranded DNA. You're going to mix it together. Some strands have a mutation, others do not. You're going to denature them with heat so they all become single-stranded, and then you're going to renature them where you'll get your mutated strands annealing back together and your normal strands annealing back together, where those are your homoduplexes. And then you're going to get some strands that have a mutation that are going to anneal with the normal strand, and those are going to be your heteroduplexes. So once you have your homo and hetero duplexes, you're going to cleave with the enzyme nuclease. So your homo duplexes are not going to be cleaved, but your heteroduplexes are going to be cleaved, because your heteroduplex has a region that are not complementary bases, so that it forms a little bubble because they're not going to attach to one another exactly, and then the nuclease is going to cleave. So when you have your heteroduplex, one strand has a mutation. When you get that cleavage, that's going to indicate that you had a mutation. So you can see that on the gel where you have your cleaved fragments are going to indicate that you had a heteroduplex that has a mutation in it. There's also something called invader technology, where you can detect mutations with an enzyme called cleavage, and this enzyme is a proprietary enzyme that's only used in this specific technology. So your sample is mixed with probes and enzymes. The enzyme cleavage is going to cleave your probe test sample hybrid and yield a fluorescent signal. So your signal is only going to be generated if the probe and the test sample sequence are complementary to one another. So here you have your invader technology where you have your probes, your enzymes, and everything is in a 96 well plate. You have your mutant probe that's going to hybridize if you have that mutation present. The wild type probe will not hybridize because you don't because you have a mutation, so the wild type is not going to match. So you're only going to get cleavage if you have your hybridization, and then you're going to get fluorescence. So in summary, chromosomes are analyzed by gene sustaining and karyotyping. Karyotyping detects changes in chromosome number and also large structural changes. More subtle chromosome changes can be detected by metaphase or interphase fluorescence in situ hybridization. Mutations and polymorphisms are changes in the DNA sequence. These DNA sequence changes have varying effects on the phenotype, and molecular detection of mutations include hybridization, sequence, or cleavage-based methodologies.